Hi, everybody. People of Section 1 in Biology 142. Here is yet another snow day recording. This is right in the middle of Chapter 17. This recording will be on the special sense of vision. So you're going to see this in the lab on Wednesday, assuming there's no snow day on Wednesday. So on Wednesday, you'll be looking at models and a dissected cow eye. These parts, these anatomical structures will come to life a lot better on those. Here in lecture, we will expand quite a bit on the physiology of the special sense of vision and some accessory structure. So grab a warm drink, put up your feet, and here comes almost an hour's worth of entertainment from Biology 142. So let me go to screen sharing and we'll get started. So this is our opening slide for special sense of vision. And this is just trying to illustrate why a physiologist or an anatomist would be immediately attracted to special sense of vision. For a physiologist, it looks like that more than half of the sensory receptors in the entire human body are located in the retina portion of the eye. That's an astonishing imbalance in favor of the special sense of vision. When an anatomist looks at the brain, a huge portion of the cerebral cortex, most of the occipital lobe, and the association areas are dedicated to receiving and processing visual information. Unheard of amount of proportions in the brain. And then when an anatomist looks at our 12 cranial nerves, 1/3 of them, 4 cranial nerves, are designated to assist in one way or another a special sense of vision. So all these facts on the ground, on the anatomical ground, indicate that we do have something special here. Indeed, special sense of vision, a very frequently used, frequently relied upon sense of detecting changes in our environment. Light and its changes bring a lot of information to us. Before we get into the actual location of those photoreceptors and how we get there, we will be looking at some accessory structures. So let's start at the visible accessory structures. You know what? Let me see if I can make this full screen. Here we go. Maybe a little better this way. So the receptors are located in this yellow layer of the eyeball, and many portions of the eyeball are essential to have physical integrity for special sense of vision to happen. So to protect the eyeball itself, we have added structures. And when you take a look at the eyelid, and including the eyebrows and eyelashes, we have physical structures to protect physical impact, debris, dust, even pathogens to get or stay permanently on the eyeball. We'll be talking about in a minute of the tear-producing apparatus, the eyelids with every blink smear a small volume of tears over the cornea here to keep it moist. Very important for the precise functioning of the special sense of vision. Same is the role of eyebrows and eyelashes, catching debris before it gets into the eye. But once a debris, dust, pathogens hit the anterior aspect of the eye, we have another ingenious structure When you look at this entire eye socket, under the eyelids, it would be very easy for pathogens or debris just to slowly sweep into the structures behind the eye and cause inflammation or some kind of a damage here. So here we have a enclosure provided by this very thin, light brown, U-shaped structure called the cornea. I'm sorry, U-shaped structure called the conjunctiva. The conjunctiva is a very thin membrane, and you may have heard this mentioned when you hear the word conjunctivitis. So, because conjunctiva forms a little cup 360 degrees around the eyeball, anything that would normally could move behind the eyeball is caught here in this little trap. And so debris pathogens tend to accumulate here until they are moved out of the eyeball through blinking and the flowing of the tears. Conjunctivitis is a redness, pink eye of this membrane, because when irritation happens, whether it's a viral infection, bacterial infection, or just scratching or itching, mechanical irritation, the blood vessels of the conjunctiva dilate, and with the pink eye, with conjunctivitis, we are seeing the color of blood in those widened blood vessels. Here is another important protective system, the tear-producing apparatus or lacrimal apparatus. Tears are liquids made by glands. Here we have the lacrimal glands dedicated to that role, and a very small volume, about 1 milliliter of tears produced per day. They are released gradually, and with every blinking, the tears are smeared over the cornea to keep it, excuse me, transparent. When we have excess amount of tears, some kind of an emotional upset or a strong irritation of the cornea or conjunctiva, tear production ramps up, and large amounts of tears needs to be taken out of the eye. So at the corner of the eye, we have lacrimal canals, lacrimal ducts that eventually take tears, excess tears, into the nasal cavity. These structures we will review in the lab. We have 6 muscles in each eye socket to move the eyeball. These are skeletal muscles. We have 4 straight or rectus muscles and 2 oblique muscles that grab onto the eyeball from the side. We will, in the lab too, associate cranial nerves with these muscles. So cranial nerve 4 is dedicated to superior oblique. We'll have a visual confirmation on this particular illustration. This doesn't make a whole lot of structural sense. Take my word for it. Cranial nerve is associated directly with superior oblique. That's the only muscle it innervates. Cranial nerve 6 is singularly associated with the lateral rectus muscle. And cranial nerve 3 brings nerve impulses to all the rest of the skeletal muscles. So this is how these muscles get their nerve impulses. If you remember way back in chapter 16, we mentioned these 3 cranial nerves as part of the cranial bulbar pathway of the direct pathway of nerve impulses to move skeletal muscles. When we look at the eyeball, we'll be doing this in the lab too. We'll explore the eyeball according to its layers. The layers are called tunics, and we're going to go from outside and dig deeper and deeper. So the first thin tunic is the fibrous tunic. It's called fibers because it has connective tissue and connective tissue fibers in it. One challenge for the eyeball is to lead light deep into our body to the bottom of the eyeball where the retina is located, which is not highlighted here. So it makes sense that we have to create some special transparent tissues. The very first of these transparent tissues is the cornea. This little bubble-shaped portion of the fibrous tunic is transparent, has a very unique connective tissue structure with it. Because damage to the cornea, whether it's embryonic malformation, trauma, heat-related denaturation, or chemical influences, scarring of the cornea, all these make visual experience less maybe a blurry vision results. We do have procedures to replace damaged corneas, mostly using transplants. Now, why is cornea so important so that we resort to surgical replacement? That's because the cornea's function is refraction. Refraction means, in plain English, focusing light. So we don't want to lose a transparent focusing portion of the light, so we can replace it surgically. If you heard about the LASIK surgery manipulates the cornea. Take a look at this here, a little thicker representation of the cornea. LASIK surgery cuts into the cornea, gives it bursts of laser, and then when the cornea heals from misshapement, the cornea is being reshaped, changing the focusing ability. We'll come back to this aqueous humor. This is this bubble of liquid deep to the cornea, and it is important to bring oxygen and nutrients to cells of the cornea. So this is the first part of the fibrous tunic, transparent, has the ability to focus the light, to refract the light. The large portion of the fibrous tunic is totally translucent to light. It blocks the movement of the light, and this is called a sclera. From the end of the cornea all the way through, it forms almost a complete sphere, cranial of two, has an opening on it. So this is the white of our eye. The function of the sclera is protection of the entire eyeball, because the toughness of this dense irregular connective tissue is identical to the dermis or to the toughness of heart valves. When we peel away the fibrous tunic, we'll come to this pinkish layer here, the vascular tunic. Distinct feature of the vascular tunic is large amount of blood vessels, hence the name vascular tunic. We'll have a number of small and somewhat unusual name structures. So let's take a look at this, again, almost completely spherical layer. This is the choroid. When you look at these cow eyes in the lab, you'll see that the choroid is dark purple in color, almost black, because it has a lot of melanin coming from melanocytes. This dark color helps to absorb scattered light inside the eyeball. The blood vessels of the choroid bring nutrients to the retina. So there are two functions associated with this layer. And as you see on the cow eye, choroid is a paper-thin layer. So don't think about a finger-thick tissue. This is extremely thin, extremely delicate. So if we continue the examination of the choroid, getting closer to the beginning of the cornea, we'll see a much thicker structure. Right here, this is the ciliary body. It has a band of smooth muscles, the ciliary muscle. This helps to change the shape of the lens. We'll be looking at this towards the end of the lecture, just how this muscle can carry out the changing in the shape of the lens. As we'll see, changing the shape of the lens is immensely important to focus on objects, to be able to see sharply objects closer than 20 feet to us. Another part of the ciliary body is ciliary process, this Swiss cheese looking portion here. And this ciliary process makes aqueous humor. I mentioned aqueous humor already. It fills up this bubble under the cornea. As we'll see this very soon, aqueous humor has another compartment that it fills in the eyeball. So these are the two major parts of ciliary body. Now, already here you see yet another portion of the vascular tunic, iris. Iris is a, at least from the outside, is a colored ring-shaped band. Right in the middle of the iris, there's an opening, the pupil, and that allows light to pass through the vascular tunic. When we talk about the iris, we will mention a number of characteristics, including its ability to regulate light that goes into the eyeball. So let's switch over to the iris and pupil slide here. The iris, the biggest function of the iris seems to be highly visual, that it gives the color to our eye. It doesn't really make any difference for the special sense of vision, whether the iris has a brown, blue, or green, or even a red color to it, because the iris's function comes from its smooth muscle. But for a minute, let's talk about eye color. If your eye color is brown, it's those dark brown pigment, melanin, is reflecting and absorbing light throughout the iris, and that's where your eye color comes from. That's the most frequent eye color. For those of you who have blue or green eyes, your iris does not have blue or green pigment in it. Blue or green eyes come from the reflection of the lights from these ridges, and you can see these ridges here. Depending on how these ridges are shaped, light is reflected as blue or green color, and that's where those two eye colors come from. Let's go back to the really life-saving aspect of iris, muscles. So we have two sets of smooth muscles. In the next slide, we'll be looking at the backside, the smooth portion of the iris, not the colored portion, and we'll see how these muscles are located. So here we go. We're looking at the backside of the iris. The lens would be right here. So we're looking at the posterior aspect of the iris. And what we see is two groups of muscles. We have the radial muscles, like the spokes of a wheel, and we have the circular muscles that surround the pupil. So let's step into bright light, and today it's really tough to find, but when the sun comes out, eventually there's enough light that our retina has to be protected from strong light. So what does the iris do? The circular muscles contract and narrow the lumen. So in bright light, the lumen, the pupil will be narrowed. When we step into a dark environment, the radial muscles of the pupil, of the iris will contract, and the pupil will be increasing in diameter. This reaction, the dilation of the pupil, can also be brought about by the fight or flight response. Sympathetic division of our nervous system can send nerve impulses to the radial muscles, and the same effect can be achieved by fight or flight response as stepping into a dark environment. In dark environment, dilation of the pupil allows more light to enter the eyeball, and therefore we have a better ability to observe our environment. So you have seen this in many illustrations now, the lens. The lens is totally avascular, meaning it has no blood vessels, meaning it is not part of the vascular tunic, right? It just wouldn't make sense. So the lens is yet another totally transparent tissue, but instead of connective tissue, what made the cornea transparent, the lens is made by the proteins called crystalline. The lens is surrounded by a capsule, and the function of the light is the function of the lens is focusing light or reflection. We'll be seeing that the image by the lens is always focused on the fovea centralis portion of the retina. The lens is also anchored by these very thin ligaments, suspensory ligaments. And another aspect of the lens is that, while it's not pictured here, the lens is surrounded by a thin capsule. This thin capsule is kind of important because let's mention another eye surgery. When the lens grows cloudy with age normally, this condition is called the cataract. So the cataract, this cloudiness of the lens, blocks light, and so the visual experience of the individual with cataracts is that it's very difficult to see the environment because not enough light is penetrating into the eyeball. So to deal with cataracts, the cataract surgery slits open the capsule, breaks up the lens inside the capsule, and suctions out the debris. And the empty capsule is filled with a glass or a plastic lens. And so with cataract surgery, the individual receives a new lens, either plastic or glass, to replace the cloudy one. We'll be coming back to these suspensory ligaments a little bit. When the suspensory ligaments are tight, as you see them here, the lens is in its flat shape, and the surrounding tissues are pulling the lens to this flat shape. Again, we'll come back to the changing in the shape of the lens, which we'll be calling the accommodation of the lens. After mentioning these structures, let's talk about what fills the eyeball. On that cow eye in the lab, you'll be seeing this vitreous humor filling the vitreous chamber. This is a very clear, crystal clear, glass-like in the jello consistency material. The anterior cavity is filled with this aqueous humor. We have two chambers. We'll be seeing this a lot better in the lab. Anterior chamber between the cornea and iris, and the posterior chamber between the iris and the lens. So these cavities, this cavity, anterior cavity, is filled with aqueous humor. Aqueous humor is, as its name suggests, just like water, very different from the jelly-like vitreous humor, which is filling the posterior cavity. important to talk about the aqueous humor because this is constantly made by the ciliary process, and it's also constantly drained away by this scleral venous sinus. So there is a disorder where this drainage point is blocked and pressure builds up inside the eyeball. This condition is called glaucoma. So glaucoma is the abnormally high pressure coming from the buildup of the aqueous humor. There are pharmaceutical-based, surgical-based treatments of glaucoma. Glaucoma is dangerous because increased pressure inside the eyeball will put pressure and blocks or collapses blood vessels on the retina, and our photoreceptors will not get enough nutrients to function fully. So glaucoma can lead to visual impairment. And this takes us to that portion, retina, which is also called nervous tunic. So this is the third layer highlighted in yellow here. We have a number of areas on the retina itself. The major features here are the photoreceptors, rods and cones. And you see the surface blood vessels coming to bring nutrients to the retina. Now the choroid provides a different supply. With glaucoma, it's these blood vessels that could be pressured to collapse. The retinal blood vessels, when they are observed during an eye exam, they can also inform the optometrist that this person may be suffering from diabetes or hypertension. In those two disorders, blood vessels are damaged and sometimes closed off. And so looking at retinal blood vessels can be very informative if anything non-eye related disorders might be happening to this patient. When it comes to retina, we'll be focusing our attention on the photoreceptors, but there are other things in the retina as well. So in this particular illustration, the light is coming from below, and the light goes through one set of neurons called the ganglion cells. This provides the ganglion cell layer. Light is then go through the bipolar cell layer, yet another group of neurons. And only after that, the light hits the photoreceptors, and we'll be seeing cones, these green ones here, and the rods. And these two photoreceptors will react to light. The reason why we will spend a good deal of time on the photoreceptors, because imagine a speeding bullet with the speed of 670 million miles per hour. That's pretty fast. That's the speed of light. And these receptors have to react to that speedy change in the environment. It's totally mind-boggling what these receptors do for us. So we will spend some time with them, explore them a number of ways. And we're also going to see how these photoreceptors will create neurotransmitters to start nerve impulses in the bipolar cell layer. So here are rods and cones, very basic characteristics we list here. Rods, you can see the shape, is a rod shape. We have over 100 million of these, and they detect shades of gray. The other receptor type are the cones. Look like a cone, right? And they give us the color vision. So high improvement in the quality of detection, this change in our environment, seeing color. An interesting aspect of cones, excuse me, that they increase their density at a particular area, macula lutea of the retina. At that point, cones are packed against each other like sardines. That's where we have the sharpest vision and the best color vision in the macula lutea portion of the eyeball. So here is how photoreceptors eventually create nerve impulses. The rods, once light hits them, they have to transduce light into electrical signals. So the rods will be releasing a neurotransmitter called glutamate, as you see spelled out here. And in dark, rods and cones release a lot of this glutamate and block nerve impulse creation of the bipolar cells. When light is around, rods and cones will slow the glutamate release, and that release from inhibition will convince the bipolar cells to create nerve impulses. These nerve impulses will be then passed down to ganglion cells, and the ganglion cells will form cranial nerve 2 and head all the way into the brain. The pathway is briefly indicated here. Cranial nerve 2 will lead to the thalamus and then to the occipital lobe, to the primary visual cortex. This is the gross anatomical exposure of all that. So the eyeballs would be right here in this area. Here is where cranial nerve 2 comes to the middle. The two optic nerves, cranial nerve 2s, meet in the middle, called the optic chiasm. This is a highly unexpected structure here. We're not going to go into analyzing what's happening here. Suffice it to say that nerve impulse moves on to the optic tract into the thalamus. You don't need to know the name of this nucleus here. And from the thalamus, the nerve impulse will go into the occipital lobe for detection and for interpretation. So what is this feature that enables the photoreceptors to interact with light? The magic ingredient is both rods and cones or photopigments. The photopigments are made up of a protein called opsin and a retinol. It's the shape of the retinol that changes when light hits it, and this change in the shape of the retinol will change the shape of the protein, and this will start changes inside the rods and cones themselves. So it's this retinol, that small organic molecule that actually catches or interacts with the light, and then larger components, proteins, the first being opsin, that will change the behavior of these receptors. For the rods, there is a single kind of photopigment, rhodopsin. For cones, there are three photopigments. We have red, green, and blue photopigments. The opsins are red, green, and blue type. So these three different kinds of cones inform us the change in the color of the light that's around us. Retinol is part of all photopigments, both in rods and in cones. So this is what's happening inside these rods and cones. And we're not going to learn all of these changes here, but It's hard to see. Here's the retinol. In dark, the retinol has its band shape, and in dark, rods and cones have a strong flow of sodium ions into the receptors. The strong flow of sodium releases a lot of glutamate, this inhibitory neurotransmitter. So bipolar cells are inhibited by this big cloud of neurotransmitters. When light hits a photoreceptor, retinol straightens out in its shape, and sodium channels will be closed. The low sodium inflow into the receptor will slow, in turn, the neurotransmitter release, and a little bit of inhibition leads to the excitation of the bipolar cells. So when we release the bipolar cells from inhibition, it comes from the lowering or slowing the glutamate release from the receptors. Rods and cones differ a little bit in their connections. Take a look at a cone here. It is connected almost one-to-one to a bipolar cell. Take a look at rods. Many rods converge on a single bipolar cell. This is called a convergence in the retina. This is why rods are more sensitive. They work in dim light because hitting any single rod from this network of 600 or so can trigger a nerve impulse. So let's talk about, just for a second, horizontal and endocrine cells. They are mixed with the bipolar cells here in the bipolar layer. This is the earliest manipulation, earliest processing, earliest change in the nerve impulses. So the nerve impulse does not have to go to the brain to be adjusted. Contrast is increased by these horizontal and endocrine cells while the nerve impulse is still going through the retina. When it comes to image formation itself, the major word is refraction. We need to have this by the cornea and the lens. We will be talking about the change in the shape of the lens because the focusing ability of the lens has to be changed. We already discussed the constriction of the pupil. We're not going to revisit that. But let's take a look at refraction first. So why is the cornea, this static portion of our eyeball, can focus light? Because when light rays hit the cornea, light coming through the air, and just by hitting this waterlogged cornea, light changes direction. And this change in direction is refraction, the focus light. This is the best way to explain why our lens needs to change shape. Take a look at the top image here. Here's a pine tree more than 20 feet away. The lens is in its relatively flat shape, and the suspensor ligaments are tight, pulling the lens to the side. Please notice that the ciliary muscles are relaxed here. This is the default workings of a lens. Muscle is relaxed, lens is relatively flat. We make perfect imagery from objects more than 20 feet away. When you look at your smartphone, suddenly this arrangement will give you a blurred vision. So how can we create sharp images from objects closer than 20 feet? Here comes the accommodation of the lens. The ciliary muscle will contract, suspensory ligaments will relax, and the lens will be more curved. I think when you compare the two images here, when the lens is flat versus curved, you can see a more bulging nature to the lens. And this increased curvature, this accommodation, is going to produce a sharp image from objects closer than 20 feet away. So this is why we have this ciliary muscle to contract when we look at objects closer than 20 feet, so the lens can be accommodated and we can see a sharp image. And the last topic here is convergence of the eyeballs themselves. This is the function of skeletal muscles that change the position of the eyeball. Our two eyeballs are highly coordinated in their movement, and they are always angled towards the middle. This angled nature is called the convergence. Again, this is brought about by skeletal muscles. The outcome of this convergence of the eye is that we gain depth perception, 3D vision, and this increases our visual senses tremendously, because seeing something in 3D is a lot more informative than seeing things just in the 2D around us. So the convergence of the eye is this medial angling of the eyeballs, and it is coordinated by those skeletal muscles that you mentioned very early on during the beginning of this lecture. So this is it for the special sense of vision. I'm going to stop sharing here. And so I hope to see you all on Wednesday. Please remember, on Wednesday we have lab all day. I'm going to send out yet another reminder. So at 11 o'clock on Wednesday, we'll be meeting in the lab. Start with that exquisitely crafted lab quiz, and then we have vision and endocrine system labs all the way till 3 o'clock. Hope to see you all on Wednesday. And if you have any questions, please send me a message. And of course, I'll be there all day on Wednesday, so happy to answer questions there. All right, I'm going to say goodbye.